Welcome to this episode of Real Chemistry. We're going to talk about the citric acid cycle today. It's one part of a bigger picture called metabolism. And what we're really going to focus on in this lesson is understanding those steps. So you might see a lot of videos where you go through all these steps really quickly and they just kind of say what happens, but they don't necessarily explain why these reactions are occurring. And so we're going to focus on that in this lesson. First up, let's kind of contextualize where we're at in the process of metabolism. Remember that metabolism is just the process of converting molecules into energy, ATP. And that we start metabolism of glucose with glycolysis, where we split glucose in half. That happens in the cytosol. And then pyruvate is formed at the end of that glycolysis, and we convert that into acetyl-CoA. So acetyl-CoA is what we're going to start the citric acid cycle with. So that's where we are now, the citric acid cycle. It's the process of taking acetyl-CoA, and eventually it's going to make FADH and NADH, as well as one ATP. Okay, so we're going to go step by step through the citric acid cycle, looking at those reactions and thinking about how it eventually produces all these energy molecules. All right, let's take a look then at the first step of the citric acid cycle. So here's our friend acetyl-CoA. I know you love acetyl-CoA at this point, and it comes into the citric acid cycle from the previous steps of metabolism. So this, if we think about what this actually is, right, check out these red carbons. These red carbons came from sugar. So the sugar that you ate, or the blood sugar that you have coursing through your veins, eventually gave these two carbons to the citric acid cycle and said, hey, get some more energy out of them. And what it does is it takes oxaloacetate. This guy just hangs out in the citric acid cycle for its whole life, constantly being used over and over again. So you can see the arrow on this side. We're going to actually end this video right back at oxaloacetate. So oxaloacetate comes along and is combined with acetyl-CoA to form citrate. So key points. The two carbons from acetyl-CoA combine with oxaloacetate to make citrate, and oxaloacetate both starts and ends that process. So this is just the very first process of putting those carbons on there. What we're going to see ultimately is that these carbons are oxidized into CO2. Remember that CO2 is one of the major products we form when we burn glucose, and ultimately your body gets rid of all the carbons that come into our body as glucose as CO2. Every one of these steps that we're going to be looking at is catalyzed by an enzyme. So they end with ACE, and usually they're going to tell us a little bit about what's going on. This one's called citrate synthase. So basically it's making citrate. Not too complicated there. All right, so where does citrate go? Well, the first thing we do with citrate is we undergo what's called an isomerization. So it just reshuffles around some stuff in the molecule. If we look at this OH group right here, right? It's on that carbon, and then in just a second, oh, it's on the bottom one. So all we did is we took that OH from the middle carbon to one below it. That's it. That's all that's happened here. It was run by the enzyme aconitase. Why did it do that? Well, here's the cool thing. If you think about this alcohol, this turns out to be a tertiary alcohol because the alpha carbon, which is right here, is connected to 1, 2, 3 carbons. Now, if you've studied the oxidation of alcohols, then you know we can't oxidize that. So this is not possible to oxidize. Can't oxidize. And our metabolism is based off of the oxidation of carbons, right? We start with C6H12O6 when we are burning glucose and we combine it with oxygen. This is oxidation. So our body wants to oxidize all the carbons in glucose, and it just does that really slowly. Now it's going to be oxidizing these remaining two carbons that have come to us through the rest of metabolism. And the way it's going to do that is by changing that tertiary alcohol to a secondary alcohol. So here, this guy is a secondary alcohol. Notice that the alpha carbon, which is right here, is connected to 1, 2 carbons. Now, you may or may not remember, but secondary alcohols can be oxidized. So what your body's really doing here is preparing citrate for oxidation. It's saying, you know what, we'd love to oxidize it. That's basically what we're doing here to all these molecules. And if I run an isomerization to move that OH there and make it a secondary alcohol, then we can do that. Okay, so step 3, what is it going to be? Well, no surprise, it's actually going ahead and oxidizing it. So what happens in step 3 is isocitrate becomes what's called alpha ketoglutarate. What we really want to focus in on here is, let me change colors, this guy right here. Notice that it goes from an alcohol to a ketone. So we add that bond to oxygen. That's an oxidation. Okay, whenever we oxidize our substrate here, whenever we oxidize anything in the citric acid cycle, you're always going to see NADH produced. Because NADH is the reduced form of NAD+. So basically we're taking NAD+ to NADH. This guy over here is the reduced form. So what's happening is we're oxidizing isocitrate so we can reduce NAD+. We give energy to that molecule in the form of electrons. So electrons have just been transferred to it. That's what reduction means. And those are high energy electrons that can later be used. Okay, at the same time, we lose a carbon. And it's good to track that because remember the ultimate products we're making in our overall metabolism reaction is CO2, water, and energy. And so our CO2s come out at various points in this process, and that's the end of the line for that carbon. That carbon is going to go to your bloodstream and be exhaled out through your lungs. Okay? Whenever you see the word dehydrogenase in the citric acid cycle, dehydrogenase, that's always going to be where NADH or FADH is formed. And that means that there's an oxidation going on of the main thing going through the citric acid cycle. So in this case, isocitrate was oxidized, and that allowed NADA plus to be reduced. Okay, so that's step 3. Step 4, what's going to happen? Well, notice here, if we look at the enzyme, we see alpha ketoglutarate dehydrogenase. Again, we see the dehydrogenase, so we know that there's going to be an oxidation going on. The main change in this molecule, and this is kind of a good skill when you're looking at these complicated reactions, is compare the molecules that we start and end with, right? We start with alpha ketoglutarate and we end with succinyl-CoA and see what changed. Okay, pause it. Try to figure out what changed. What changed is down here we have C double bond O. That goes off as a CO2. And now we instead have what's called coenzyme A at the end. So this guy has come into our citric acid cycle and attached to our alpha ketoglutarate. That turns out to be an oxidation because the atom attached in the coenzyme is a sulfur. So whenever we oxidize something, we increase the number of bonds to oxygen. Sulfur turns out to act the same way. So increasing the bonds to sulfur also oxidizes something. So our molecule is being oxidized, and once again, just like we'd expect, we reduce NAD+. So we get a NADH, which will later be turned in for energy. Okay, now what happens from there? Well, next up, our coenzyme A is popped off, and this turns out to be a high energy molecule over here. And it's a low energy molecule over here. And so what happens is that energy, the energy difference between those two molecules is dumped into ATP. Okay? And at the same time, our coenzyme A is just popped off. Okay, so that's step 5. It uses succinyl-CoA synthetase, which means we're making succinate. Where do we go from here? Well, once again, we'll see that what we do is we oxidize our molecule. This time we do it in a slightly different way. Notice if I look at succinate and I compare that to fumarate, there's just one small difference. Again, pause the video, try to find it. What happens is a hydrogen's been removed. So there were 2 hydrogens on each of those carbons, now there's 1. What that means is there's a double bond here. Okay, so we formed a double bond. So 2 hydrogens are lost. And that's an oxidation process. When we lose hydrogens, it's also oxidation. Where do they go? Those 2 hydrogens go right on to FAD and make FADH2. That's where the 2 hydrogens go. So we've once again reduced an energy-carrying molecule to make FADH2. Okay? Notice that the enzyme used is succinate dehydrogenase. So we see that dehydrogenase again. And whenever we see that, we know that there's an oxidation step going on. That our main thing in the citric acid cycle is being oxidized, which is always going to reduce either NAD+ or FAD. Okay, next step. Here our body does something interesting. It takes fumarate and it remembers that, hey, if I have a double bond here, that's an alkene, and I add a water to it, that actually is a hydration reaction where we get out an alcohol. So this is a reaction you could easily look at in organic chemistry where you're just taking a alkene to an alcohol. Why would your body do that? What is it interested in? Well, what it wants to do is create the molecule in such a form that it can be oxidized again. And now notice we have an alcohol. And it is a secondary alcohol, so it can be oxidized. So what do you think is going to happen in the next step? That's right, it's going to be oxidized. So step 8 is the oxidation of malate. And when it's oxidized, then we once again reduce NAD+ to NADH. So we make another NADH. The enzyme is malate. What do you think the second part of that name is? It's dehydrogenase, right? Because remember, whenever we are oxidizing our molecule in the citric acid cycle, we are using a dehydrogenase. So alcohol is oxidized and it goes ahead and reduces the NAD plus to NADH, and oxaloacetate is reformed. Remember, this is where we started. So we're to the end of the citric acid cycle, and now this can just repeat. Oxaloacetate can grab another acetyl-CoA. So here's a summary. Our citric acid cycle overall produced 1 ATP, 3 NADH, and 1 FADH, all energy-carrying molecules we care about. The carbons in acetyl-CoA are oxidized repeatedly to form NADH and FADH2. So that's really what's going on in the citric acid cycle is oxidation. It prepares the molecule for oxidation, it oxidizes it. It prepares the molecule for oxidation, it oxidizes it. Eventually, all the carbons in acetyl-CoA are converted into CO2. So we got out 2 CO2, and that's how many carbons were on our acetyl-CoA. And the cycle occurs twice for each glucose you put in. So we haven't mentioned that, but if you think about the fact that our acetyl-CoA came from pyruvate, which came from glucose. Let's kind of track that for a second. Glucose went into glycolysis and became 2 pyruvates. So it was split in half. Pyruvate has 3 carbons each, and then pyruvate gets converted into acetyl-CoA. And it gets converted into 2 acetyl-CoA. Now that 2 acetyl-CoA can be plugged into the citric acid cycle one time for each acetyl-CoA. Okay, so that's a summary of the citric acid cycle.